Meiosis is a very specialized form of cell division, whereas mitosis is used for pretty much any type of cell within the body. Meiosis is going to be specific for the sex cells or gametes. In humans, that turns out as the eggs and the sperm. The reason for this is because in these gametes, we need to do two things. First, we need to have half the normal number of chromosomes. If we didn't have this halving, every generation our genetic material would double, and within a few generations, the amount of genetic material per cell would be simply unsustainable. The other major event that's going to happen during meiosis is going to be genetic variation. You're going to get, we'll talk about two different forms of genetic variation that we'll get during meiosis. So meiosis actually has two stages. If you remember during Biology 111, meiosis 1 is going to start off with a prophase 1, metaphase 1, anaphase 1, and telophase 1. So we're going to go through similar steps as we did through mitosis. But again, there are some major significant differences between mitosis and meiosis. So your book goes into quite a lot of details about what's happening during prophase 1, different subphases and so forth. But the major thing that I want you to be aware of is that during prophase 1, you're going to have homologous chromosomes finding one another, they're going to pair, and they're going to exchange bits of genetic information. During metaphase 1, the major difference between mitosis is that during metaphase 1, these homologous pairs are going to align in the center of the cell instead of single file like they do during mitosis. And then you can see here, this pair of chromosomes, they've exchanged little bits of genetic information through the synapses that happened during prophase 1. During anaphase 1, you're going to get separation of the pairs of chromosomes. The homologous chromosomes will separate, and they are going to move towards opposite sides of the cell. During telophase 1, the daughter cells will now have one chromosome from each of the homologous pairs. Which one they get is going to be totally random. So the cells are going to reform their nucleus, they're going to separate, they're going to have cytokinesis. You are then going to have a resting phase or interkinesis. During this phase, unlike interphase, the cells will not duplicate their genetic material. During meiosis II, you're also going to have a prophase, a metaphase, an anaphase, and a telophase. But there are some important differences between both meiosis I and mitosis. So during prophase 1, you're going to have a vesiculation or disappearance of the nuclear envelope. You're going to have formation of the spindle fibers and so on. During metaphase 2, the chromosomes now will align single file at the metaphase plate. During anaphase 2, you're now going to have separation of the daughter chromosomes. So those sister chromatids, one half's going to one side of the cell, one half is going to the other side of the cell. During telophase 2, the spindle fibers disappear, the nucleus reforms, and you're going to have some cytokinesis happening. The end result of meiosis is that you are going to end up with 4 cells instead of the 2 cells that you get from mitosis. Also, these cells are going to be haploid, or have half the normal number of chromosomes. In humans, the sex cells, meaning the eggs and the sperm, are going to have 23 chromosomes, so that when the egg and the sperm unite, you will have a total of 46 chromosomes, which is what we see in all of our rest of our cells. So let's look a little bit more at some of the genetic variation that we get through meiosis. The first is crossing over. So what happens during crossing over is that you're going to have the sister chromatids from one pair of the homologous chromosomes and sister chromatids from another homologous pair of the chromosomes. There's going to be some crossing over that's going to happen. They're going to exchange bits of genetic information. And at the end of this, you're going to have 4 different potential daughter chromosomes. This is going to greatly increase the genetic variability of the offspring that an individual might be able to have. So this crossing over, don't forget, that happens during prophase 1 of meiosis 1. independent assortment happens during metaphase 1 of meiosis I. What happens is the homologous pairs, when they line up at the center of the cell, the order in which they line up is totally random. So you could have all of the chromosomes that originated from the father on one side and from the mother on the other side, or they could be mixed up, and you can see with just 3 chromosomes all of the different possibilities. And now you can imagine with the 23 pairs of chromosomes that we have in humans that we have a very, very large number of different possible combinations of offspring. You combine that with the genetic variation that we get from the crossing over, and it is numerically impossible to have 2 identical siblings. Now, the only exception, of course, is identical twins, and they're a special case that we will talk about later. So here are a few questions. We will quiz ourselves. So during gametogenesis, we have a difference in our production of sperm versus eggs. So the production of sperm is called spermatogenesis, and the production of eggs is called oogenesis. During spermatogenesis, you have one primary spermatocyte. This cell is going to go through meiosis I, and it's going to go through meiosis II. After maturation, you can potentially have 4 functional sperm from your one initial starting cell. In oogenesis, in contrast, the egg is going to be much, much larger than the sperm cell. It is also going to have the majority of the cytoplasm, the majority of the organelles, and so forth that the developing zygote will need. So what happens is, when these cells divide, they do not divide equally. Instead, most of the cytoplasm and the stuff goes to one of the cells. The other cell is known as the polar body. So you can see here after meiosis I, there's an unequal division. Majority of it is going to go to this particular cell here. After meiosis II, there is a second polar body that's formed, again because of unequal division. So at the end of this oogenesis, what's going to happen is you're going to only have 1 functional egg instead of the 4 functional sperm that you get through spermatogenesis.